Hello, my students. I'm your biology teacher, Cik Govin. Our topic today is sexual reproductions in flowering plants, and our subtopic is 6.3 Pollinations and Fertilizations. Learning standards. Learning standard number 1, describe pollinations. Number 2, describe the formations of pollen tube and the formations of male gametes. Number 3, explain double fertilizations in the formations of diploid zygotes and triploid nucleus. And number 4, justify the importance of double fertilizations for the survival of flowering plants. Pollinations and fertilizations. The process in which pollen grains are transferred from the anther to the stigma is known as pollinations. This process is assisted by pollinating agents such as insects, mammals, birds, water, or winds. The presence of pollen grains on the stigma triggers the process of fertilization. This diagram shows the pollinating agents, birds, bees, butterflies, winds, water, and beds. Formations of pollen tube and male gametes. Look at these diagrams. This diagram shows the pollen grains. Okay, this is anther, the male reproductive organs, and this one is stigma, the female reproductive organs, and this one is the pollen grains. Okay, the matured pollen grains. We look at number 1. The wall of anther from matured pollens will dry, shrinks, and splits. The pollen grains in the pollen sac are released, released from the anther. And number 2, the released pollen grains are transferred to the stigma of the same flower or different flower by pollinating agents. Okay, number 3, the pollen grains that have been transferred to the stigma will germinate and forms a pollen tube. The pollen tube grows down toward the ovule through the styles. Number 4, the generative nucleus will move along the pollen tubes towards the ovules. At the same time, the generative nucleus will divide by mitosis to form two male gametes. Okay, when the pollen grains attach to the stigma of the female reproductive organs, the pollen grain will germinate to form the pollen tubes, and then the generative nucleus divide by mitosis to form two male gametes. Okay. Stage number 5. The end of the pollen tube will secrete an enzymes to digest the tissue of the styles. Okay, when the pollen grains forms the pollen tube will growing downwards through the style by secrete an enzymes to digest the tissue. So when it reaches to the embryo sac, the pollen tube will penetrate the ovules through the microfile. This is microfile, the openings of the embryo sac. The tube nucleus will degenerate and both male gametes enter the embryo sac. Okay, now we look at what is double fertilizations in the formations of diploid zygote and triploid nucleus. Double fertilization involves two male gametes in which the first male gamete fertilizes the egg cells. Okay, you look at this one. There are two male gametes here. One fertilizes the egg cell. This is the egg cells, and the second male gamete fuse with the polar nuclei to form a triploid endosperm tissue. This is the polar nuclei. Okay, one fused with the egg cell, another one fused with the two polar nuclei to form a triploid endosperm tissue. As discussed previously, the generative nucleus undergoes mitosis in the pollen tubes to produce two male gametes which are haploid. Thus, both male gametes will enter the embryo sac for fertilization. So these two male gametes are from the generative nucleus. Okay, generative nucleus divide by mitosis to form two male gametes. Okay, number one, when it reaches the embryo sac, the pollen tube will penetrate the ovules through the microphiles. The tube nucleus will degenerate. Okay, the tube nucleus will generate. Okay, and then both male gametes enter the embryo sac. And then number two, one of the male gametes fertilize the egg cell and produce a diploid zygote. One of the male gametes fused with the egg cell become diploid zygote. Okay, this is diploid zygotes. And then the second male gametes fused with the two polar nuclei to form a triploid nucleus. Okay, triploid endosperm nucleus. This is the triploid endosperm nucleus, and this one become the diploid zygotes. The importance of double fertilizations for the survival of flowering plants. Number one, the fusions of one of the male gametes with the egg cell produce zygote. This ensures that the genetic information is passed down from one generation to the next and restore haploid conditions in gametes with the formations of a diploid zygote. Number two, the fusion of another male gamete with the two polar nuclei produce endosperm tissue. This tissue is used for the development of an embryo for the survival of plant species. In eudicots such as legumes, mangoes, and mustard, the endosperm is fully utilized by the embryo to develop before the seed matures. In most monocots such as coconut, wheat, barley, and corn, only a part of the endosperm is utilized for the development of an embryo. Some of them are stored in the cotyledons to be utilized during the germination of the seed. The endosperm tissue enables the embryo to survive in the seed for a long time if the conditions are not favorable for germinations to occur. Activity 6.4. The aim of this activity is to carry out an activity to observe the germinations of pollen grains and the formations of pollen tubes in a sugar solutions under a light microscope. The materials: 10% sucrose solutions, distilled water, acetocarmine stain solutions, fresh flower such as balsam, hibiscus or alamanda, and filter paper. The apparatus: cavity glass slide, cover slips, forceps, mounting needles, dropper, and light microscopes. The procedure: number one, prepare a clean cavity glass slide. Okay, this is the cavity glass slide. Using a dropper, okay, this is dropper. Place 2 drops of 10% sucrose solution onto the surface of the slide. Okay, 2 drops of 10% into the cavity of the glass slide. Take a fresh flower with enter that has many pollen grains. Okay, so make sure when you take enter from any flower, make sure have many pollen grains. And then number 3, dip the enter into the drop of 10% sucrose solutions that has been placed in the cavity of the slide here. Okay. 4. Procedure number 4. Cover the specimen with the cover slip gently using the mounting needles. Okay. Needle to prevent the formations of air bubbles. 5. Observe the slide under the light microscope using low power objective lens to obtain the preliminary observations on the shape of the pollen grain. Sketch your observations. Leave the slide in the dark place at room temperature for 20 minutes, and then once again observe the slide under a light microscope using low power objective lens to observe the pollen grains that has germinates and forms pollen tubes. Number 9, place a drop of acetocarmine stains solutions on one end of the cover slips on the slide. By placing a filter paper at the opposite ends, let the stain solution diffuse across the cover slip to stain the specimen underneath. Once again, using the slide that has been stained under the light microscope, observe the tube nucleus and generative nucleus in the pollen tubes. Draw and label a diagram to show the pollen grains before and after the formations of the pollen tubes. Okay. Discussion question number 1. What is the need to use fresh flower in these experiments? If we use fresh flower, we obtain fresh pollen grains so that the germination process can occur faster. Number 2, a group of students did not follow the correct procedure. They did not use the 10% sucrose solution. Instead, they used distilled water. Predict the result obtained by them. Okay, if they use distilled water, the formations of pollen grain do not occur because germination process require energy supplied by sucrose. Okay, when we use water, mitochondria cannot generate energy, okay, because lacks of sucrose. Number 3, another group of students forgot to stain the specimen by using the acetocarmine stain solutions. State the problems they may have. So if they not stain the pollen grains, so the pollen tube is difficult to observe because the pollen tube is colorless. Okay. Formative practice 6.3. Question number 1. What is meant by pollinations? Pollination is the transfer of pollen grains from the anther to the stigma. Anther is the male reproductive organs and stigma is the female reproductive organs. Question number 2. Pollination is very important to ensure that reproductions in flowering plants occur successfully. Explains. Pollinations transfer pollen grains from anther to stigma. The presence of pollen grains on the stigma initiates the fertilization. So without pollinations, there is no fertilization. So pollinating agents is very important to ensure that the fertilizations occur. Question number 3. During the formations of pollen grains, the microspore mother cell divide by meiosis and produce tetrad. State the feature of the tetrad and the importance of such feature. Tetrad cells are haploid to maintain the number of chromosomes in the zygotes after fertilization. Okay, meiosis is important to ensure that the number of chromosomes is maintained from one generation to the next generation. Question number 4. The generative nucleus divides by mitosis in the pollen tubes. State the importance of this process. Okay, it produces two nuclei, namely the tube nucleus that will form the pollen tube and the generative nucleus that will form the male gametes. Okay, there are two male gametes produced by process of mitosis in generative nucleus. Question number 5. The megaspore cells contain 8 nuclei. How does it occur? The nucleus of megaspore mother cell will undergo mitosis 3 times to produce 8 nuclei. Question number 6. State the role of double fertilization in ensuring the survival of flowering plants. The fertilizations of male and female gamete produce a diploid zygotes and transmit genetic information to the next generations. the fusions of the second male gamete with the polar nuclei to form a triploid endosperm nucleus that will develop into the endosperm tissue. This tissue supply food for seed germinations. Okay, that's all for today. See you in next lessons. Thank you and bye-bye.